Okay, hello everyone. Welcome to this week's C3 AI DTI seminar. My name is Manxi. I'm co-hosting this seminar series with Professor Sastry and Professor Srikant, who are the panelists today. C3 AI DTI Transformation Institute has the mission to support science and technology developments that address the challenges and support the leading scientists in the field of academia, industry, and society. And this weekly seminar series on every Thursday features some of the recent research that happened in exciting field that study how artificial intelligence and machine learning tools can lead to scientific breakthroughs with large-scale societal benefits. I want to pause here a bit to thank the contributions from our academic and industry partners. This semester series has a special focus on incentive design and learning for societal systems. And here is the list of incoming speakers who have confirmed their talks for this semester. They are all very exciting, and you can find more information about these speakers as well as the title and abstracts of their talks on our on the website. So we have had 4 seminars in the past 4 weeks. We posted the videos and slides on our website as well as the YouTube channel. So you can watch them later if you have missed it. The easiest way to follow us and make sure you don't miss our event is to subscribe to the newsletter as well as our YouTube channel. Today, it is our great pleasure to have Professor Christos Papadimitriou here to talk about his work titled as How Does the Brain Beget the Mind? Professor Papadimitriou is the Donovan Family Professor of Computer Science at Columbia University. Before joining Columbia University in 2017, he was a professor at UC Berkeley for the previous 22 years. And before that, he taught at Harvard, MIT, NTU, Athens, Stanford, and UCSD. He has written 5 textbooks and many articles on algorithms and complexity, as well as their various applications in a broad range of fields. He holds a PhD from Princeton and 8 honorary doctorates from top universities around the world. He is a member of the National Academy of Sciences of the US, the American Academy of Arts and Sciences, and the National Academy of Engineering. And he has received the Knus Prize, the Godel Prize, the von Neumann Medal, as well as the 2018 Harvey Prize by Technium. He has also written 3 novels titled as Turing, Logicomics, and his latest novel called Independence. So this seminar will be his talk for 45 to 50 minutes. And for the audience, please use the Q&A feature to submit your questions, and we will ask them at the end of the talk on your behalf. So without further ado, Christos, the floor is yours. You can start sharing your screen. Thank you.
Thank you. Many thanks, Manxi. Many thanks for inviting me in this wonderful seminar. I just want to start by pointing out that what is happening now is incredibly mysterious and miraculous. I know for a fact that none of us understands how it's done. What I mean is that I create airwaves, sound waves in my space. These airwaves are reproduced in yours, and your brain takes these sound waves and makes them words, and then creates hierarchies of these words that we call them sentences, arguments, stories, and things like that, narratives, and stores them away. And if I'm very lucky, you are going to remember them for the rest of your lives. And how is this done? Okay, there is zero doubt that this is done by neurons and synapses, but all of us know a little about neurons and synapses. I had to learn a lot recently. And we know that seems impossible. How could this happen in neurons and synapses? And this is what I'm going to explain in this talk. I'm going to tell you sort of in terms of a story, because I had my brain was opened by a bunch of encounters and especially by experiments. Okay, so what I'm going to show you now next is a very clever experiment that opened my eyes. So what they did at NYU, they played a tape, a very monotonous tape, where single syllable words were told at 4 hertz, and they recorded they repeated this for 5 languages and many hundreds of subjects, and the tape was something like this, single syllable words. And then what they did is they took all the data, all the recordings, they passed them through the discrete Fourier transform, and of course what happened is that there was a peak at 4 hertz. Then they did something clever. They repeated the same experiment, except every 4 words made sense. And guess what happened? There were 4 peaks at 4 hertz, because 4 times every second you have to recognize a word, but also 1 hertz. What happens once a second? Once a second is a sentence is formed. And what happens twice a second? A phrase is formed, as in bad cats eat fish. Okay? So here is what I think happens. Your brain builds hierarchies. If this was a longer story, there would be a taller hierarchy. So this is incredible. Okay, so I don't know. So I at this point I like to take a poll, but think about it. Do you believe that your brain makes trees? I think you should. I asked Chris Manning at some point. I said, Chris, do you believe that we make trees in our brain? And Manning thought and said yes. Okay, so and I chose Chris Manning because I was told that he was among the least likely people to admit that our brain does trees. We make trees in our brain. How can this be done by neurons? Okay, how are these 3 representations created? And they have to be created about 12 spikes per step. Why 12 spikes? Because brain activity in this context is about 50 hertz, and language is 4 hertz. So with those spikes, you have to make progress in building these trees. And we are nowhere close to understanding how this is done. The last few months, we are a little closer, and this is what I'm telling you. So how does the language organ work? When you are faced with an impossible problem, a very interesting and common maneuver is to generalize it, okay? And ask, how does the mind, in other words, cognitive, this is a famous cognitive science textbook, in other words, cognitive phenomena, language, intelligence, art, math, and so on, planning, emerge from the brain, from molecules, neurons, synapses, from wet stuff. And there is a huge gap between the two. Okay, there's a gap of scale, of course, but also a gap of scientific culture, emphasis, and of course types of animal subjects used. So this man is my idol in this field, Richard Axel, my colleague at Columbia, Nobel Prize 2004. And 4 years ago, he said this: we do not have a logic for the transformation of neural activity into thought. I view discerning this logic as the most important future direction of neuroscience. Okay, so there are, let me point out, I find this extremely surprising. One surprise is that Axel uses the term logic. which to many of us means a formal system, ultimately computational. And I agree, he was 100% right there. The second surprising thing is that 4 years later, I don't know anybody else who works on this, okay? So, but I'm going to tell you the progress I have made on that. I have been working on this problem for a few years before Richard posed it in this, articulated in this beautiful way. So here is what I'm after. What kind of formal computational theory would qualify as Axel's logic? Okay, and where does computation happen? Okay, it happens in molecules in the brain. It happens in spiking neurons and synapses. And those who know about the brain, they will tell you that the real computation happens in the input device of the neurons, the dendrites. But I mean, no, it also happens in whole brain. I mean, cognitive scientists, what they do is they make experiments and then they look at the data and they say, you see, it's as if the subject executed, the subject's brain executed this program. Okay, there is also whole brain computation, but there's a gap in the middle. And this is the gap that I want to fill by something that is called assemblies of neurons. What are assemblies of neurons? The old timers among us remember that in the 1970s and 80s, there was the neuroscientists were trying to convince us that there is something called the grandmother cell. And this means that, there is a cell in our brain, so that the spiking of this cell is coterminous to thinking of our grandmother, okay? And as decades went on, things changed, okay? First of all, these are called the Jennifer Aniston cells, okay, more than one cell, because we've learned that every neuron has about between 1,000 and 10,000 incoming other synapses. And as a result, if grandmother is one neuron, then there is no grandmother, okay? But in order to have a grandmother, she must occupy tens of thousands of neurons, okay? So, and they did look for cells recognizing familiar people like Jennifer Aniston in the 90s, and in her honor they called those Jennifer Aniston cells, even though Travolta was also involved. These population of cells, because you see what you do when you do an experiment, you have a subject, you record from a few dozens of their neurons, and you show a couple of hundred pictures, and you get a hit. You can't be so lucky, okay? So because you chose a few dozen out of 10 million neurons, and you show 200 pictures, and you got a hit. This means that every familiar picture is represented by at least 10,000, maybe 30,000 neurons. And these are called assemblies, these ensembles or collective memories of neurons. Hebb, or Donald Hebb, many years ago, first proposed them. And neuroscience was looking for them for 5 decades in vain, until improved technology in Busaki's lab found them in the act. Sort of, they saw them spike together to recognize the concepts. And by now, so my colleague Rafa Juste at Columbia basically creates them in mice, ignites them, makes them represent a behavior like clicking, and then he presses a button, so a laser light shines on a neuron, and the mouse clicks. Okay, what is an assembly? Assembly is a large and densely interconnected set of excitatory neurons in a brain area, whose firing in a particular pattern is tantamount to our thinking of a particular memory, concept, person, name, word, episode, and so on. Okay. In his recent popular book, Gyori Busaki, who is a leader in this field, calls them the alphabet of the brain. So assemblies is not something that I pull out of thin air. It's a very obvious, well-known reality in the brain that is well recognized and seen. So about a year and a half ago, I wrote a paper with my friends called "The Brain Computation by Assemblies of Neurons." And I mean, if you want to go back and understand better some of the things that I've seen, this is the paper, and in fact, figure 3 of this paper, figure 2 of this paper, that explains better than I can what's going on and why assemblies are the thing to watch. So in this paper, we articulate what we call the assembly hypothesis, that there is an intermediate level of brain computation, lower than cognition, higher than neurons, and it is implicated in higher cognitive functions, such as reasoning, planning, language, storytelling, math, science, music, the good stuff. And assemblies of neurons are its basic representation, its data type. Okay, the question is, of course, what are the operations? And projection, association, merge, pattern, complete, and then control commands like read has an assembly fired in this brain area. Fire, make this assembly spike. Inhibit and disinhibit areas and fibers. So this is the whole language, the whole programming language, and it's basically the basis is what I believe, what I'm proposing is Axel's logic. Okay, so I'm going to of all these operations, the most basic that holds the key to understand the key to understanding what assemblies are and how they work is the projection operation. In fact, it's so basic that this is how assemblies are created. Okay, so I'm going to say a little in more detail what happens here. Basically, what you have is you have an assembly in one brain area on the left. There is a red arrow, which means there is afferent computation. In other words, there's outgoing computation to the right area. And in the right area, there's also recurrent computation. In other words, the recurrent synapses. Okay, there are afferent synaptic connections, the red arrow. In other words, there are synapses going from left to right. A lot of random synapses, you can think of them. Then there are a lot of random synapses recurrently in the right area. And there's also inhibition. That's the purple stuff. And Hebbian plasticity. In other words, if a neuron appears to make another neuron fire, then their weight increases, let's say, by 10% or 5% or something like that. So this is the whole model. And based on this model, we predict the following. Okay, so you can prove it mathematically that will happen with high probability, where the probability, of course, is the random graph underlying. That once this fires, and by the way, it's a circle, but remember that it is 30,000 excitatory neurons all around the area on the left. And then this creates another 30,000 neurons because of the inhibition, the purple stuff, because the inhibition does not allow it to fire more. And this new set of neurons fires together with the previous set, and because now there is a recurrent computation in the situation, the equations of motion of this dynamical system have changed. You get a slightly different, not slightly, usually the overlap is about 50%, set of neurons, and you repeat that, okay? And basically, eventually, the question is, does this process converge after a dozen or so steps? And is the result densely connected? And will it respond to a new firing of the stimulus? In other words, we want the new thing to be a copy of the old one. And every time the old one fires, we want the new one to fire. Will it pattern complete? This means if you light it up in the end, if you light up, let's say, if you fire 5% of its neurons, will 97% of its neurons fire? And is stimulus overlap preserved? This means the following, that if you had two such stimuli and you created two such copies, it's of course of interest, the overlap of stimuli, which basically corresponds to their co-occurrence relationship correlation, should also be preserved. And the answer throughout is yes, but sort of, I will have to tell you the details of the mathematical model. So my talk contains something that you never see in talks about the brain, a mathematical model of the brain. Okay, in fact, this is a simulator that is public and you can play with it. Okay, so you know the starting command is load brain, which is a great thing to do to type in the morning. So we have a finite number of brain regions. Okay, the compartments in blue. Each contains n neurons, for simplicity the same number, and inhibition means that only k of them fire at any moment. Some pairs of areas are connected by fibers, by bipartite random graphs, and all are recurrently connected by random graphs, by Erdős-Rényi graphs. Neurons fire in discrete steps. The k neurons with the highest synaptic input are selected to fire in each area. Okay, this is called random project and cap, RPNC, and it is the basic operation of this model. And this is an important primitive which models the inhibition excitation equilibrium in an area, which is, so all these things are basic tenets of neuroscience, which I have sort of stylized mathematically. Connections between areas can be inhibited, disinhibited, and then there is Hebbian plasticity. So, and there's also other things that I'm not bothering you with. And the main parameters of the model, I'm thinking of n in the millions, many millions. I'm thinking of k in the thousands. The probability of connection, let's say, is 0.1%, and the plasticity increases weights by 5% for each file. So, and the theorem is that all the answers is yes. Okay, are yes for that the projection converges exponentially fast with high probability. So this is not a theorem. This is like theorems from 5 papers, more and more accurate and parsimonious parameters, which show that yes. So you know, but we didn't stop there. We simulated in our model, and we simulated with realistic neurons. Okay, so there are many aspects with which all these operations, but essentially the projection do work. Okay, that they do represent a honest, accurate modeling of what can happen in the brain. Okay, so I know what's in your mind. What do neuroscientists think about this model? In general, they like it. Discrete time steps are unrealistic, but are not distortive, and we also do it in more realistic asynchronous models with STTP. The random project and cap operation is a natural simplification of the excitatory inhibitory balance. So here are the caveats, the sort of the warnings. Assemblies and plasticity are well known, well understood, but have not been thought in previous work at this time scale. 2 to 4 hertz. Okay, so this comes as a slight surprise. Oh, really? So you have them work so fast? Okay, you know, because assemblies were considered usually more stable things. I mean, no, there is no data that says they're not. Probably an assembly should not be confined in a single area. Okay, so you know, this is a very there are some very convenient mathematical simplifications that we did not avoid the temptation to make. Okay, you know, but in general, this is a rather honest mathematical model of the brain. And instead, which part of the brain? Okay, you probably know that 95% of all work in neuroscience is about the senses. Okay, sensory and motor cortex. For a change, I want to focus on the white stuff in this picture, okay? Which is 50% in the sheep, but it's 95% in the human, okay? And that's the association cortex. It is everything that is not neither motion nor senses, okay? It is basically where brain has its inner life, okay? So a rat's brain does not have much inner life, but human brain, look at it on top. It only has inner life, okay? So you know, and the senses are afterthoughts, okay? So it's time, I thought, that we should. And by the way, what connects the two? What is the apex in the middle that connects the two? That's the hippocampus. Okay, so all right. So let me see how we're doing the time. Okay, so association of two assemblies. That's the second operation. Okay, I'm going to present it because it's another beautiful experiment which again sort of moved me. So basically, what happens is that if you have two stimuli that have already on the left, which have already created two copies, two assemblies, a representation on the right. If these two stimuli now co-occur, okay, they fire together. As a result, these two assemblies are going to shed some cells and borrow some cells from each other, and as a result, overlap. Okay, to record the fact that these two stimuli co-occurred. Okay, so here's an amazing experiment. Okay, they recorded from many people, many neurons, many images, but let's think about sort of one image, one neuron, one person. Okay, and that's the Eiffel Tower, and they found the Eiffel Tower neuron in this person. Okay, they saw other famous places, nothing. They saw famous people, nothing. Then they did the following clever thing. They photoshopped Obama in front of the Eiffel Tower. And of course, beautifully, the neuron sees the Eiffel Tower and fires. Then they saw the Eiffel Tower by himself. Of course, it's going to fire. Other places, nothing. They saw Obama. Guess what? Believe it or not, the neuron fired. Okay? My voice breaks when I say this, okay? But this poor person learned that Obama has been to the Eiffel Tower, and you found out experimentally, okay? Wow. This is something, okay? This operation is called association. So projection is how assemblies are born and propagated from the hippocampus to the association cortex to enact brain computation and cognitive phenomena, okay? This is my theory. Association creates a web of meaningfully overlapping memories and possibly an orthogonal probabilistic genre of brain computation, which I haven't explored. So what I want to emphasize is that assembly operations, this language is not something that sort of, like people in the 60s said, okay, should it have a loop or should it have only conditional branching? Okay, it's not. I didn't pull it out. I didn't pull these operations and data structures out of thin air. Okay, they correspond to behaviors of assemblies that have been observed in experiments or are strongly suggested by other experiments. I'm going to talk about that. They can be provably compiled down to the activity of neurons and synapses. Okay, so in other words, we know that these operations can be implemented by neurons and synapses. Okay, and we have shown that by mathematical proofs or simulations in our model, and by simulations in systems of neurorealistic spiking asynchronous neurons. So that's the assembly calculus. And if you ask how powerful is the system, it turns out that it can perform with high probability arbitrary n over k space computations. If you are a computational theorist and you remember that n over k is in the thousands, this is a lot of computation. Basically, it says anything that can be done in n parallel steps by Boolean operations can be done here. Okay, so this is an incredible amount of computation, and I don't think that the brain harnesses it all. Incidentally, pattern completion, which is the other thing that I showed you, is this. Okay, that it's a beautiful experiment by Rafa Juste. He created an assembly that basically instigates a mouse to lick and get sugary water, but if it sees some pattern, but if it does not see the pattern, but Juste fires these lasers into two of these neurons, the red neurons, the rest will fire and the mouse will lick. Okay, so assemblies are not science fiction. Okay, so they are everyday things at Juste's lab. Okay. So here's what I'm saying. This is the gap that I started with. I'm telling you that it can be bridged by assemblies of neurons and their operations. And what we have to prove is that it must be implemented by spiking neurons and synapses. It can, check. And now I must show you that it implements cognitive phenomena. Okay, and for this we're going to go back to language. Go back to language. Okay. How, we have known for 150 years that there is Wernicke's area where word choice is made. There is a lexicon, we know now, where all words are stored in all languages. There is Broca's area where syntax seems to be happening. We know this from lesions for about 150 years. But how? What? When? And why study language? Okay, so here's my argument. Axel thinks that it's crazy to study language. Okay, so I think it's crazy not to, because it's a last-minute adaptation. 3,000 generations ago, about 1% of the split from the 0.5% of the split from the chimpanzee, we acquired language, and language in this 1% evolved to exploit the brain strength. So it. Language is a mirror of our brain and its capabilities. Okay, and it's an incredibly opportunity loss if we don't study it. Okay, and in fact, we have some incredible experiments. I showed you a few. Here is another one. They read to subjects sentences like the ball hit the track and the track hit the ball, and different areas of the superior temporal gyrus. That's the green region on the left in the picture I showed you. Responded to track in the two sentences. Okay, this means that apparently our brain, our language organ, has a different place to store object and different place to store subject. Okay, and all these experiments and many more, so probably many hundreds of experiments, are sort of recounted and explained in this beautiful book by Angela Frederici, basically, we start to understand how things work when we hear words. Assemblies in our lexicon are excited. They seem, then something happens in Wernicke's area in response to that, and eventually Broca's area is mobilized. Okay, but I mean, as she was developing this narrative in page 134 of her amazing book, Angela sort of seemed incredibly frustrated in a paragraph, okay? And she says, I hypothesize something that can be called language mirror cells. Mirror cells are some very special cells that we have and make us repeat what we see other animals doing, emotions, usually gestures. But I mean, and if you read carefully the sentence, Angela is praying for assemblies. Okay, so and when I sent her my paper, she embraced it, and we have written a paper since then explaining why Angela why this is what Angela needed to complete the understanding of syntax, at least. Okay, so and now I'm going to present you an experiment that we made. And this is the last part of my talk. It will not take me 15 minutes. But those of you who thought that were disbelieving and thought that this was high-flying theories, pay some attention here. Okay, so we designed a parser of English written in the assembly calculus. In other words, we designed a program that gets us its input sentences and creates a parse tree. Okay, and that's not very impressive, because over the past decades, amazing programs like that have been written. Okay, what is special about this program? That it has no variables, no if then else, no loops. Its execution consists only of spiking of neurons. Okay, nothing else. So it's only spikes of stylized neurons. I want you to absorb this, okay? So what is the architecture? There are, it basically reflects the part of the brain, the language organ that I showed you. It is a brain area that we call lex, where it has assemblies, okay? By the way, this is not how we learn language, okay? I am explaining what I believe happens when we read to an adult who already has this in place, to a healthy adult, language healthy adult who has this in place. When we read a sentence like a cat chased a dog, okay? So there is this area lex. That's the lexicon that students of language in the brain believe exists somewhere in the medial temporal lobe. And then it has sort of the Wernicke's area on the left, which is subdivided into some areas, okay? We need about a dozen areas. I've shown you here verb, subject, adjective, prepositional phrase, or object, okay? And there are fibers that connect these areas, okay? Fibers, remember, are bipartite graphs, dense bipartite graphs of neurons that connect random things on the left to, back and forth. So, random neurons to random neurons in both directions. Okay, so that's architecture. The fibers can be inhibited and disinhibited individually. Lex contains an assembly for each word, and each word assembly has an action set. What's an action set? It's inhibit or disinhibit commands, a short sequence. And this action set is important because it encodes the word's part of speech, its syntactic role. In other words, all nouns have the same action set, or adverbs have the same action set, or prepositions have the same action set, okay? And when the word assembly fires, its action set is executed. So the sum total of word action sets, that's the grammar, okay? In other words, in this model, the grammar is captured precisely by the action sets of all, in a more complete model of distribution of our action set, if you wish, for each word. So what happens on input cats chase dogs? Okay, in the beginning, it starts in the initial configuration, in the initial state of this device, where only the fiber that connects lex to subject is disinhibited. Okay, why is that? The reason is because this is an English parser, and we know that the subject will come before the object. Okay, in other languages, this would not be the initial state. Okay, so this baby, this adult, as baby, learned these rules. Okay, so for parsing sentences. So it starts this way, and then cats is heard or read, and as a result, the assembly for cats fires. Since this is disinhibited, this will create, let's look at the assembly for cats. It has semantic neurons, of which plural neurons is one, phonetic neurons, which helps us, how do you call it, and recognize the word, and action neurons, okay, which encodes its part of speech, okay? So these action neurons are shared by all nouns, okay? These plural neurons are shared by all plural nouns, okay? And maybe plural verbs, and so on. So this is, and after this is done, this fiber, the top fiber is inhibited, and a new fiber that connects subject with verb is disinhibited, because now we're looking for a verb. Many things will happen in between. In this simple case, the verb comes next. So the verb comes next, chase. It opens immediately the fiber from verb to verb, from lex to verb, and it projects itself over there. Once it finishes the projection, it does some other things, like it disinhibits the fiber to object, because Chase knows that it is a transitive verb, and therefore an object is to be expected. And then similarly, it disinhibits the fiber from lex to object, and eventually will disinhibit the verb lex to verb. And finally, dogs comes. It's projected because the fiber to object is disinhibited. Finally, we go back to our initial state. And the point is the following, that after input cat chase dogs, and assume there is a full stop there that basically reinitializes everything, assembly synaptic connections, connecting synapses between assembly and Wernicke's area, form a valid dependency tree, a valid parsing tree of this sentence. Okay, and this will be repeated in many much more complicated sentences with 30 words. Okay, so this is how because so far it's a fair question. How do you know that it parsed? Okay, this is how we know that it parsed. Excellent. So it's written in a slight extension of the assembly calculus, where we add assemblies with actions. And I'm repeating, the parser is implemented exclusively through the spikes of stylized spiking neurons, okay, nothing else, okay? There is no parser like that before. In my opinion, there is no program like that existed before. After processing a sentence, it leaves behind a retrievable trail of side effects called the parse. The prototype is running. The code is available. The paper has appeared, in the Transactions of the Association for Computational Linguistics, and it's available on Archive. So, an important consideration in the assembly calculus is the following. Okay, you are telling me that you are going to write programs with this. How is an 800-line program designed, stored in the brain? What is the scheduler that is executing it? What's going on, okay? So these aspects might also be plausible. And the parser is streamlined. Each input word excites the corresponding assembly, which affects changing of the state of the device, and on and on and on. So this is, in other words, this use of the assembly calculus, which this is the use that should be legislated, is very close to a data flow language, okay, and execution environment. All right, so it's really driven by the input, okay, by what happens in the outside world. So it parses simple sentences such as the young couple in the next house saw the old little white car of the main suspect quite clearly. Okay, so that it will parse it, no problem. So we have parsed thousands of sentences like that. Speed is about 20 or 25 spikes per word, which is approximately the speed I'm talking about. I'm talking to you right now. So if you translate it into sort of human brain speed, that's what it is. And more features can be added very easily, but with much more work, we can do sentence embedding. We're doing it right now. Polysemy, ambiguity, backtracking. So sentence embedding means, for example, the young couple who lives in the next house, the parser would see a second verb and would scream. Okay, so so you need a lot of extra work to handle embedded sentences. Polysemy, of course, that means that your initial attempt at parsing may be wrong. Okay, at every moment you have to guess what this parse tree is going to be. Okay, so so we don't face this, but we know how to do it. And we also have a simple Russian parser, which is interesting because the Russian parser has very different areas, and it has the same set of fibers but it has very different areas and used in a very different way than the English parser. And presumably, both Russian kids and British kids are and American kids are born with the same brain. Okay, so how on earth do Russian kids have have their own wiring of the language organ, and so these are all amazing. I mean, my point is the following: that a bold experiment, okay, so entitles you to ask questions that were heretofore unaskable. Okay. Once we have this model, and in the absence of competition, okay, we are entitled to ask, can this be the neural basis of language? This is something that Chomsky has been talking about for 70 years. Is this it? Is this what we mean by the neural basis of language? An area that is able to accommodate assemblies for words with action sets, connected with fibers. And we know that there are evolutionarily novel fibers in this neighborhood, with Wernicke's area and Wernicke's area subdivided into sub areas. So can this be the neural base of language? And if so, how do babies populate it? Okay, so in the first 3 years of their life, after 2 years old, they start naming the areas on the left. Okay, so the designated the green areas. I mean, sample question: do all babies have the same order, the same topology of these named areas, or some babies have subject and object back and forth, okay, between them? Okay, so these are questions that are sort of mind-boggling. So this is the main experiment I wanted to show you. But we are doing other things. So we know now how assemblies learn and predict. So that the assemblies can be agents of learning. We have a paper in the upcoming AI, triple AI, planning by assemblies. In other words, we revisited Terry Winograd's Block's world and showed that a very simple assemblies program can basically take as input a configuration of blocks, another configuration, imagine, and come up with a plan, or if you wish, a sequence of robotic commands for transforming the former block to the latter, okay? And reasoning by assemblies, which is something that we're thinking about right now. So, wrapping up, as many of you know, I've studied a lot of fields in my, I was fortunate to do game theory, economics, control theory, evolution, and so biology and so on. So no cliff is as steep as this one. Okay, so this is the hardest thing that I've been doing the last 8 years, the hardest thing I've done, but it's also the most fascinating and rewarding. Okay, I believe that assemblies and their operations are very good candidates, and the only one right now, to fill the gap between synapses and cognitive operations. And I described to you in some detail the Parser experiment, which basically brought us very close to really hard questions, and heretofore unaskable, as I said. So, are assemblies the seat of Axel's logic? And how do you test, verify, falsify the assembly hypothesis? Okay, so the test, verify, falsify sort of program, I don't quite believe in it. Okay, so it's sort of a pauperism, and Kuhn came later and showed us that things are not that simple. Okay, I believe that theories are not verified or falsified. Okay, theories are either tested by a few people and adopted by even fewer, and then they die natural death, or they excite more people to work on them, and they convince people, and then they become scientific reality. Okay, and what I have to do is I have to sort of continue doing the work showing what assemblies can do that are sort of plausible cognitive operations. But I want to invite you to try also. Okay, so these are my collaborators, Santosh Vempala at Georgia Tech. We started this trip 8 years ago. On the way, we were joined by Mike Collins, my colleague at Columbia, who now went to Google, and Damit Ropolsky, a brilliant student who we co-advised with Mike. Okay, that's it, and thank you very much. And
thank you. Thank you. So there are some questions from the audience.
Sure.
The first is from Carlo. Shouldn't the x-axis be labeled as 1/Hz, more hierarchical, leading to lower frequencies? I think this is referring to one of the graph in the early part of our talk.
I think it's 1, 2, and 4 Hertz. Words are 4 Hertz because they happen 4 times every second. Sentences are 1 Hertz because they happen. I'm pretty sure. So my electrical engineer is rusty, but I'm pretty sure this is right.
Okay. And another question from Siamak. You keep mentioning excitatory neurons. What about the inhibitory ones?
Okay, so the main operation that we have, you're absolutely right to ask this question. So I'm sorry I didn't make it as clear as I should have. Our main operation, which is random project and cap, is a shorthand for the interaction between excitatory neurons and inhibitory neurons. What happens that afferent synaptic current creates an excitation in an area. This excitation grows. By being excited, it wakes up the inhibitory neurons. The inhibitory neurons are much faster. They have much faster, much smaller time constant. So they start working hard, and they raise the membrane potential of the excitatory neurons so that only a few of them end up firing. Okay, and this happens very fast. So we truncate this and we say that out of all neurons who have been received any synaptic input, only the highest ones, the K highest ones, will survive. Okay, and this is how we model excitatory inhibitory balance. Okay, but thanks for your question. I'm happy to repeat this as often as you want.
Okay, thank you. Another question from Edward. The field of reservoir computing suggests that random interconnections in the brain may be just as effective as learned ones. Could this explain why one brain can have both a Russian parser and an English parser? Perhaps no two brains with English parsers have similar interconnections at all.
Yeah. So I mean, that question is very much to the point. Many of us, in theory, AI, and so on. So we are very well trained in the Erdős-Rényi model. Okay, so what's amazing about this model is that let's take the 1,000.1 graph. Okay, it has 100 million edges and so on. So 1,000 nodes. And 100,000 edges and 1,000 nodes. So I have never seen this graph, but I know everything about it. Okay, so in other words, randomness makes everything extremely precise and regimented. Okay, and this is this is what the assembly calculus is a thing that because we believe that there are reasons to believe that synaptic connections are random, not exactly Erdős-Rényi, we know that. So but actually the deviations from Erdős-Rényi are in directions that favor the arguments that we make. Okay, so but still they help you create some certainty about the outcome of this computation. Okay. So in some sense, we are trying to do the chip trick. The trick with the chip is that they found ways to insulate wires so that they can have either many electrons or no electrons, okay? And that's how they created the bit, okay? So to create the assembly, you need sort of, a similar trick, okay? So, and that comes from random graph theory.
Thank you. Another question from Gaussy. The thermodynamic efficiency of brain computation in many orders magnitude superior to that of our digital computers. Does your mathematical model provide any clues on how such gap in physical efficiency might emerge? Equivalently, how can CS architectures learn from assembly calculus?
Yeah. So, I mean, frankly, I've been thinking about this nonstop for a very long time. I can believe that assembly computations can be much more efficient than ordinary computations. I have not pinned down. So, we have not worked hard enough on the thermodynamic efficiency of those. Okay. However, they create a computational system that, to me, to my eyes, seems a little awkward. Okay, so I have not, with a straight face, presented this to an audience or a funding agency, sort of, as a possible solution. But I mean, also, I have been so busy thinking about other things that I have not thought enough about it.
Thank you.
But thanks for.
Yeah, another question from Venkat. The neural infrastructure of the brain is sitting in an ambient environment. Can you comment on the current consensus on whether this ambient environment might play a role in brain operation, distinct from spikes along the neurons?
Oh, okay. I mean, I don't like to gripe, okay? So, but I mean, this is, so everything plays an incredible role, okay? So, so I'm sure that, for example, how sort of, how blood, how blood comes into the brain, sort of, how, everything, the environment, so the cranium, the vibrations from outside the cranium, so everything is incredibly important, I'm sure, about the brain. Okay, so one of the burdens of working into the brain is that very often you meet people, you talk to people that you respect. We have unlimited respect for, and they tell you something like, Christos, if you don't take into account the cerebellum, you are wasting your time. So I mean, or the thalamus, or the basal ganglia, or so there are 1,000 things that you have to ignore if you want to make any progress, and this will make mad 1,000 people. Okay, that's.
Thank you. Are there questions from the panelists?
No, I just wanted to say thank you for a great talk. Fantastic talk. Yeah.
Thank you.
Okay.
Yeah.
Okay, thank you.
Very happy to be here.
Thank you. Yeah, I think we're getting to the time of 2. So let's thank Christos again for this wonderful talk.
Thank you.
And thanks everyone for coming today. Thank you.
Bye.
Bye-bye.